So, in the beginning of the last century, Mendel's factor, which are the hereditary units which are transmitted from one generation to another, came to be known as the gene. This term was termed by the scientist W. Johansen in 1909. The gene was regarded as a unit of function, a unit of mutation, and a unit of recombination. The experiments of the scientists George W. Beadle and Edward L. Tatum in the early 1940s on the red bread mold, which is scientifically called as the Neurospora crassa, led to the proposition of this theory known as the one gene, one enzyme hypothesis, which states that only one gene controls the production of a single enzyme. In 1957, Seymour Benzer coined the term cistron, recon, and muton, where cistron defines a unit of function, recon defines a unit of recombination, and muton defines the function of mutation. So, when we discuss about these things, we have to realize that the characters of a species are transmitted without much change from one generation to another. The inheritance patterns of characters indicates that their expression is controlled by genes which are located on chromosomes and constitute the total genomic makeup of the organism. A gene is a unit of information that directs the activity of the cell or organism during its lifetime. It passes its message along to the progeny when the cell or organism divides or reproduces. The inheritance pattern of characters indicates that their expression is controlled by genes, which are located on chromosomes and constitute total genomic makeup of an organism. Chromosomes are made up of macromolecules of two biological components, proteins and deoxyribonucleic acid, which is also known as the DNA. Genetic information is stored in nucleic acids of the cell. So, how does scientists start realizing that deoxyribonucleic acid, that is the DNA, is the hereditary substance that is important in the transmission of genetic substances from one generation to another? There is the direct evidences and there is the indirect evidences. So, discussing about the direct evidences for DNA as a genetic material, the most conclusive evidences in support of DNA as a genetic material came from the transformation of bacteria, which were the experiments conducted by Frederick Griffith on the Diplococcus pneumoniae. And another experiment that really revolutionized this concept was the mode of infection of bacteriophages experiment conducted by Hershey and Chase on the T2 bacteriophage in 1952. We will discuss about this a bit later. Discussing about the indirect evidences for DNA as genetic material, we have the first is the location of DNA in the chromosomes. Now the second point is the quantity of DNA found in a diploid cell is approximately twice that of the DNA found in a haploid gene cell. The third point, the DNA from cells of widely differing species is less alike in composition than the DNA from closely related species. So basically, the DNA from organism within the same species has similar composition. Point number 4. Among the chemical and physical agents known to alter the chemical structure of DNA without killing the organisms are those which cause mutations. Point number 5. The wavelengths of UV light that causes a high incidence of mutation closely corresponds to the wavelength absorbed by DNA. Now, sixth point is DNA replicates exactly and exhibits functional specificity. So, during the whole debate of whether DNA is the genetic material, a new experiment in 1957 by the German scientist Heinz Frenkel-Konrad and B. Singer demonstrated that RNA is also a genetic material in certain cases. In this case, they experimented with the tobacco mosaic virus, also known as the TMV virus. So, coming to the topic of nucleic acids. So the definition of a nucleic acid is, nucleic acid constitute the genetic material of all living organisms. In general, nucleic acids are large polymeric molecules composed of repeated units of nucleotides, which in turn are composed of three components: a nitrogenous base, a purine or a pyrimidine base, a pentose sugar, and phosphoric acid. Nucleotides are bound together by sugar phosphate linkages. There are two types of nucleic acid depending upon the pentose they contain. Those containing ribose are called ribonucleic acid, which is the RNA, and those containing deoxyribose are known as deoxyribonucleic acid, which is the DNA. DNA is found in chromosomes, in plastids, and in mitochondria. RNA, however, is found in chromosomes, nuclei, and cytoplasm. In cytoplasm, they exist in the form of tRNA or transfer RNA, and they exist in ribosomes as ribosomal and messenger RNA. They are also found in plastids and mitochondria as well. DNA is a blueprint that directs the destiny of a cell during its lifetime. So, when it was proved that RNA is the genetic material, there were a lot of debate going on what the structure of a DNA will be, because understanding the structure of DNA will open the gateways to understanding how they replicate and how they propagate. Now I come to the topic of DNA model as proposed by Watson and Crick, with precise information and application of Chargaff's rule. So, what is the Chargaff's rule? So, the Chargaff's rule, also known as the base pair rule, states that DNA from any cell of any organism should have a 1-to-1 ratio of pyrimidine and purine bases, and more specifically that the amount of guanine should be equal to cytosine and the amount of adenine should be equal to the thymine. This pattern is found in both strands of the DNA. So in simpler terms, it basically says that the components of the complementary pair should be in 1-to-1 ratio. In 1953, an American geneticist, James D. Watson, and an English physicist, Francis H. C. Crick, worked together at the Cavendish Laboratory of Cambridge University, proposed a model of DNA structure as a double helix form. They inferred that in each DNA molecule there are two long and parallel polynucleotide chains helically coiled around the same axis. The right-handed helices are held together by their bases, which are paired together by covalent hydrogen bonds. Purine of one polynucleotide chain pairs with the pyrimidine of the other, adenine with thymine, and guanine with cytosine. Both the chains of a DNA molecule are thus complementary to each other. The distance between base pairs is 3.4 angstroms or 0.34 nanometers, and there are 10 bases per turn of the double helix. The two helices are coiled in such a way that they cannot be separated without unwinding. The backbone of the helix is a chain of sugars and phosphates alternating with each other. The two chains of a helix are of opposite polarity, so this is defined as an anti-parallel structure. So when you're looking at the structure, if one chain runs in the 3' to the 5' direction, the other will run in a 5' to 3' direction. The width of the helix is 20 angstroms. For unraveling the structure of the DNA, Watson, Crick, and another scientist known as Wilkins were awarded the Nobel Prize in 1962. We discussed the structure of DNA so far. Now let's discuss the structure of its cousin, the RNA. So RNA is normally a single-stranded structure. So far, there are three types of RNA that have been identified that vary in size and function. The largest RNA is found in ribosomes and is known as the ribosomal RNA, or in short form, the rRNA. There is also the messenger RNA, which is smaller and is produced in the nucleus and carries the information for the synthesis of proteins, which is shortened as the mRNA. For each protein, there is a specific mRNA. Transfer RNA, also known as the tRNA, is a much smaller molecule and it collects amino acids from the cytoplasm for protein synthesis. Replication of DNA. So on replication, each DNA molecule gives rise to two DNA molecules, identical to each other as well as to the parent molecule. So, as I said earlier, there were huge debates about and different hypotheses discussing how a DNA replicates. So the first theory was the conservative replication. So what the conservative replication says that is the original double helix serves as a template. The original molecule is preserved intact and an entirely new double-stranded molecule is synthesized. The second hypothesis was the dispersive replication. So what the dispersive replication discusses is that the original molecule is broken into fragments. Each fragment serves as a template for the synthesis of complementary fragments, and finally two new molecules are formed which consist of both the old and the new fragments. Now the third and the correct replication of DNA hypothesis was that the semi-conservative replication. So Watson and Crick again in 1953 proposed a semi-conservative mechanism of replication on the basis of their model of the DNA molecule. They suggested that the two polynucleotide strands of DNA molecule untwist and start separating at one end. The separated single strands then act as templates for the synthesis of new strands. Two complete DNA double helices are formed, which are identical to each other and to the parent molecule. Each daughter double helix carries or conserves one polynucleotide strand from the parent molecule and one newly assembled strand. In 1957, Matthew Messelson and Franken Stahl at the California Institute of Technology did experiments on the culture of the E. coli bacterium to confirm the semi-conservative nature of DNA replication. The Messelson and Stahl experiment. So in this experiment, they grew E. coli bacterium. They grew two cultures of E. coli bacterium for many generations in separate medium. So there was the heavy culture and there was the light culture. So the heavy culture was grown in a medium in which the nitrogen source was ammonium chloride NH4Cl containing the heavy isotope N15, and the light culture was grown in a medium in which the nitrogen source was a more common light isotopes N14. At the end of the growth, they observed that the bacterial DNA in the heavy culture contains only N15 and the light culture only N14. Then they analyzed the two types of bacterial DNA with a technique called cesium chloride density gradient centrifugation. In this process, heavy and light DNA extracted from cells in the two cultures were settled into two distinct and separate bands. In another experiment, they transferred the heavy culture containing N15 into a medium that had only N14, leaving the bacteria in this light medium long enough to allow two replications of the DNA, which is two generations of bacteria. After the first replication, they extracted DNA and subjected it to density gradient centrifugation. The DNA settled into a band that was intermediate in position between the previously determined heavy and light bands. After the second replication, they again extracted DNA samples and this time found the DNA settling into two bands, one at the light band position and one at the intermediate position. This result confirms Watson and Crick's semi-conservative replication hypothesis. After the first replication, there were only intermediate molecules containing both N14 and N15. So basically, when the N15 DNA came in interaction with the N14 DNA and we saw the semi-conservative replication, there is one strand from the N15 and one strand from the N14, and hence we got a DNA molecule which was part N15 based and part N14 based, hence the band was in the middle. And in the second generation that we saw that there weren't heavy bands but there were intermediate bands and lighter bands, which again supports the theory that with semi-conservative replication, from generation to generation the molecules get lighter and lighter. The central dogma, expression of genetic information. The history of the analysis of gene function dates back to 1902, when an English physician, Archibald Garrett, reported that some hereditary human diseases are caused by certain biochemical defects. In 1909, he suggested that the diseases inherited from the parent to the offspring are termed as inborn errors of metabolism. The disease is caused by the failure of an enzyme to catalyze a reaction in the metabolic pathway. One of the metabolic pathways studied by him was the conversion of phenylalanine to other molecules. The metabolic pathway that he mentioned was phenylalanine gets transformed into tyrosine, which also gets transformed into melanin. This tyrosine gets converted into homogentic acid, which gets transformed into fumaric acid and acetoacetic acid. 3 human diseases are caused by failure within this pathway. Phenylketonuria, also termed as PKU, develops when the amino acid phenylalanine is not converted into the amino acid tyrosine, and the excessive phenylalanine that accumulates damages brains and other body cells. Albinism is a condition in which melanin is not formed, and people with this disease have no pigment in their skin, hair, and eyes. Alkaptonuria develops when homogentic acid is not broken down into fumaric acid and acetoacetic acid. Homogentic acid is then excreted in the urine rather than metabolized. Its presence causes urine to turn black. In phenylketonuria, the enzyme that converts phenylalanine to tyrosine is not functioning. In albinism, there is a failure of the enzyme that converts tyrosine to melanin, and in alkaptonuria, the enzyme that catabolizes homogentic acid is not working. Garrett speculated that the cells of the patient with one of these diseases are unable to synthesize the enzyme needed for the metabolic pathway. The enzyme is not formed at all or is not formed correctly because of the defect in the genetic material of that patient. Thus, the error in metabolism is an inborn or an inherited error. So with such revelations came a lot of questions regarding the central dogma of life. And one such explanation was the Beadle and Tatum's one gene, one enzyme hypothesis. The experiments by the scientist George Beadle and Edward Tatum in early 1940s on Neurospora crassa, the red bread mold, led them to propose the one gene, one enzyme hypothesis, which states that one gene synthesizes one enzyme which is responsible for controlling one biochemical reaction. They got a Nobel Prize for their work in 1958. The contributions of Beadle and Tatum laid foundation for biochemical engineering. Another such hypothesis was the one gene, one polypeptide hypothesis. All enzymes are protein molecules and proteins are complex molecules composed of one or more polypeptide chains. Thus, the one gene, one enzyme hypothesis was later extended to include all polymers made of amino acids and was called the one gene, one polypeptide hypothesis. Now, we recognize that genes provide instructions for building nucleic acids, the RNA and the tRNA, as well as polymers from amino acids, and so the modern statements of what a gene does is the one functional product hypothesis. And with that, we come to the end of the video. I hope you all understood the concepts. Have a great day, and this is Shibu signing off.